let's start the meeting. A very warm welcome to all of you for joining this BioNanoNet Association Members Welcome Webinar. Today we have the pleasure to have 3 international organizations as the speakers, and the beginning will be made by IVL, which is located in Sweden. The speaker is, in this case, John MUNTHE. He is the Vice President of Research at IVL, which is the Swedish Environmental Research Institute. The good thing is we do have a project together. It's called IRIS. And the even better thing is that we have John now here to present to all of us what we can get from IVL and how we can collaborate with you. So John, looking forward to your speech, and the floor is yours.
Thank you very much, and I will try to share my screen first of all. Yes, that looks like it worked. So here we go, and thank you very much for the opportunity to give a brief introduction to our institute. IVL, Swedish Environmental Research Institute, we like to call ourselves Sweden's leading organization for applied environmental and sustainability research. I will start with a few basic facts. Down at the bottom, you can see that we're a limited company, but we are owned by a foundation that was formed by an industry and government initiative already in 1967. The purpose then was to solve the sort of newly discovered environmental problems from industry, particularly pulp and paper, energy, etc., who were the backbone of our industry sectors in Sweden, but also causing a lot of problems. So this was a very early start and very much focused on solving problems and applied research and consulting. We have now, since then, broadened into many areas of environmental research and also, of course, sustainability these days. We have a vision called saying that a sustainable society, very simple. That sort of describes that we have a very broad areas of activities covering, as I said, almost all industrial sectors and all major environmental issues, and again becoming more and more active in social science, economics, etc., to cover the full sustainability arena. We are based in Sweden, of course. That's where we have our major operations, about 400 employees in total. We have activities in China since many years, started out as a cooperation on water treatment. And we nowadays also have employees and activities in India, again working with wastewater treatment. We have facilities in Sweden that we use in our research. Apart from people and computers and models, etc., we have chemical laboratories. We have test sites for wastewater treatment. We have and for marine research and innovation and water management, just to give a few examples. Shifting now, you can see this is a homemade slide, so not made by our professional communicators, but by me. But I wanted to say a few words about what we are doing in the field of chemicals and chemical safety. First of all, we do have a strong background in what I call environmental chemistry, that is being out in the field, sampling and analyzing for chemicals, hazardous chemicals that shouldn't be there, also trying to determine the effects, but also going backwards, finding sources and potentially the solutions to the problems. We work a lot with circular economy use and circular economy and sustainable resource use, and linked to that also, of course, hazardous substances in products and materials. If we move to the left, we provide a lot of regulatory support to industry. That's more on the consulting side than the research, but it's an important part. And we work with the whole life cycle from production to waste to products and waste and recycling, and we have a large group working with life cycle assessment and various models and methods for sustainability assessments, including risks from hazardous chemicals. And it all boils down to the bottom where it says safe and sustainable by design, which you all know is a relatively new concept, but incorporates many of the headings on the other circles there. What I would like to do now is to show a few examples of projects we are running in Sweden and internationally, just to give you an idea of what our activities look like. I would like to start with MISTRA SafeChem. It's a large national research program focusing on green and sustainable chemical industry. It's funded by the MISTRA, the Swedish Foundation for Strategic Environmental Research, called MISTRA. You see all our research partners and our industry partners here at the bottom. And what is this? This is a research program, as I said, targeting green and sustainable chemistry with a holistic approach to chemicals. We try to cover the whole value chain. The main research areas are new synthesis processes, tools for improved risk assessment and life cycle management, and the whole idea is to predict and avoid risks already in a design phase. It's led by IVL. We have 5 other research partners and 12 industry partners. It's funded by MISTRA, as I mentioned, about 70 million Swedish crowns over 4 years. That's 6.7 million euros. And the partners contribute with additionally 30 million Swedish crowns. It was granted in 2019, started summer of 22, and the first phase runs to 2024. And a few more details about what we are doing in the project, IVL and the research partners. Starting at the top left, innovative industrial production processes. In our case, this means enzyme and metal catalysis to a large extent. Can we replace traditional chemical production methods with these? That will solve a lot of problems and move towards less waste and zero and less environmental impacts. I won't go through them all, I don't think there's time, but hazard and risk screening for early warning and proactivity is something important. Life cycle assessment, materials management, green chemistry education, etc. We do work with case studies so that we can move from, let's say, basic research to applied research and test them in real environments quickly. And we are aiming at a common vision and establishing a platform for research on green and sustainable chemistry in Sweden. And this is just a picture showing the very simplified picture of the value chain of issues of areas where we are working, from renewable carbon feedstocks to design for safety, sustainability and recycling, moving along the value chains. And to a large extent, we are developing the tools we think are needed to enable the shift to a green and sustainable chemical industry here. Of course, we have a website. You are very welcome to follow it and sign up for our newsletters. We also are active on LinkedIn, and we off and on arrange conferences and webinars within this program. The second example I would like to mention is IRIS, already mentioned, and I think many of you who are here are familiar with this. So I will probably do this quite quickly. A newly started EU project with this scope to connect, synergize and transform the SSBD community in Europe and globally towards the life cycle thinking. Again, it's a project very much focusing on building, sharing and transferring skills and knowledge, and to develop a permanent network, very much focusing on implementation of the SSBD linked to of course, to the EU chemical strategy. And we have also the ambition to establish more cooperation in this area with the industry, with the European Commission, and of course with research performers around Europe. A long-term goal of this project is to build a structure for continuous cooperation and services to network members. This is something we think will be highly needed with the chemical strategy being implemented, and in particular the criteria for safe and sustainable by design, to start building a training services for companies, for SMEs, and services for knowledge exchange and sharing that will be needed for a successful implementation. So this is something we really believe in. You see the consortium here, and if you are not already involved but want information, I urge you to contact my colleague Emma Strömberg. You see the email address there. I think she's also in this meeting, so she can read if you have a message in the chat. And eventually there will be websites, etc., and communication activities from IRIS. So I think this is one of the more interesting projects we are running right now. Another example I wanted to give, which is not really research, but it comes from research we have performed. This is a subsidiary we have, Basta. It's a company owned by us and the building industry. And the focus is to provide a system to phase out harmful and hazardous substances in construction products, Basta Online. And it's in reality a searchable database with 170,000 environmentally evaluated products, where we have, with the company, has evaluated and assessed the contents of hazardous substances in these products. So if you are a builder wanting to buy construction supplies and products, you can use this database to search for and find suppliers of these materials that have been evaluated and checked for contents of hazardous materials according to a procedure developed by Basta. I wanted to show this because I think it's interesting to see how we can turn research results into something that is useful for a longer time, since even after the research programs have ended, and for the, specifically in this case, for the construction sector. We were also asked to say something. Why are we interested in BioNanoNet? Why I want to be associated with BioNanoNet? Of course, BNN is a partner in IRIS, but we see opportunities to increase our knowledge exchange and dialogue on safe and sustainable chemicals, materials, and products. Contribute with our own knowledge and long experience in research on safe chemicals, green chemicals, life cycle perspectives for materials, and also circular solutions. To find new research partners and new opportunities for research and innovation in the field of safe and sustainable chemicals and materials. And more practically in the short term is to explore upcoming opportunities in Horizon Europe in the next phase, 2023-2024. And with that, I will end with just showing you the address to our website, ivl.se, and of course my email if you have further questions or want to be in touch with us. And I think we have 1 or 2 minutes to, if there are any specific questions right now. Thank you.
Thank you very much, John. Very interesting things you can offer here, and I'm sure there will be lots of good contacts following your presentation. I would have one question about the Basta Online, which might be also very useful for others to hear about. How far is it open that other can contribute data, for example, or that they can, let's say, connect with this? What is the strategy? Is there any opportunity to collaborate there?
I think what is it's built up as a system where the suppliers have a strong responsibility for providing information on their products and ensuring that they do not contain hazardous substances. And then we have a system of independent auditors. And I think to some extent, the data is limited to, let's say, regulated compounds, and there is a discussion, always discussion going on, whether to move on into risk substances that are not regulated, and so. It's very much, it's not the BASTA company in itself do not handle a lot of data on the hazardous properties. That is done by the companies themselves. So, and they use various public information sources, of course, and it's also a system where it's not completely open about information on what the products contain. That is not possible under for the market conditions. So that's why we have a system of independent auditors to check. And if you have more questions, I'd be happy to put you up in contact with the people working with BASTA. I am not personally involved, but I think for me it's an interesting example how to take research into reality.
Thank you very much. There comes a question from Blanca from TEMAS. Are there any industrial associations in IRIS? As far as I heard, it was an academic-based initiative, is her question.
Maybe I can ask Emma to answer that, but there are a strong industrial participation in ASTA, in IRIS. Please, Emma, you know much more better than I do.
Thank you, John. Yes, we do have a very strong connection within IRIS with several industrial associations. We have 6 different value chains included in the project, and of course, very strong collaboration with SADEGHI as well.
Thank you, Emma, and nice to see you again.
Good to see you.
Perfect. In sake of time, it's perfectly to move forward. Thank you, John, once again.
Thank you.
And I would kindly ask to Blanca to open. Yeah, here she is. Video. Nice to see you. Blanca MERINO. She's co-founder of the TEMAS Solution GmbH, a company located in Switzerland, and of course active in entire Europe and maybe even globally. It's a pleasure to have you back here in the family. We are happy that we can welcome you now with your new affiliation, and I'm sure the people will be very interested in the things you can offer from TEMAS Solution. So Blanca, the floor is yours.
Thank you very much for giving us the opportunity to introduce our new company called TEMAS Solutions. Indeed, my name is Blanca MERINO. I am a co-founder of this company. So very swiftly, just to remind everyone what Andreas just mentioned, we are a small company which was founded in August 2020. We are very young, but we are very privileged to have highly experienced staff among us that cover the different areas of knowledge that we do. And in a nutshell, our main goal is to bridge the gap between innovation or innovative ideas into regulatory compliant products. And to do that, and I take over from the previous presenter, we do follow a safe and now sustainable by design principle. This is our main aim in a nutshell. We cover different areas. This is a very busy slide just to give you a flavor of the different topics we cover, which goes from environmental solutions to, for example, water treatment using nanotechnologies all the way. I'm sorry, I don't know why it's moving automatically. Anyway, going all the way to regulatory affairs and risk assessment, and even we provide also financial analysis and business plans. So yeah, we can now dwell a little bit further into any of these topics. But before we do that, I just wanted to introduce us here. As I said, we are quite small. We are 5 people in the company. From left to right, you can see our colleague Devendra Joshi, and he's our business development and IT. And below we have a Dr. Veronique Adam. She's our environmental risk assessor coming from EMPA, from Bernd Novak's group. She's been working in the environmental risk issues for many years. Then we also have our colleague, Dr. Beatrice Salieri. She's our sustainability consultant, also coming from EMPA with several years of experience. Then our colleague, Dr. James Baker. He's very experienced in the international project management as well as development of remediation solutions. For example, in water treatment issues. And myself, I'm Blanca SUAREZ. I'm a toxicologist and risk assessor with several years experience in nanosafety issues. So one by one, I will take you through our different services. Regarding sustainability assessment, we try to help companies now comply with the Green Deal goals. Unfortunately, I cannot show you any particular projects because we work under contract from companies, so I cannot share with you particular projects. Nonetheless, I'll take you through different services that we offer. We provide life cycle assessments and also strategies to manage carbon footprint for companies. We also work on sustainable strategies in different sectors like chemicals, emerging technologies, and packaging. As an example here, I showed you that we are deeply involved in the development of nanotechnology solutions to address current environmental problems. For example, to remove contaminants for water treatment, contaminants like arsenic, fluorine, or boron. And you can see the different icons below here. We follow the Green Deal and the Sustainable Development Goals from the United Nations. Now, regarding safety and regulations, we produce the regulatory dossiers, again, for the sectors that I previously mentioned, chemicals, cosmetics, and medical devices. And while doing so, we help companies identify any regulatory data gaps that they may have and they need to compile the successful dossier. And for this, we also have a large network of GLP compliant testing labs, which we offer to our customers. Below here, you see the different icons regarding the regulations we follow. We are fully aware of the medical device regulation and the European Union. We also have the cosmetic regulations under our skin, as well as the REACH regulation, which deals with chemicals. And we are also leading a project under the graphene flagship called SafeGraph, where we are producing a regulatory roadmap for graphene-based products, and we will release this roadmap in the autumn next year, 2023. Now, regarding safe and sustainable by design, it was already mentioned by the previous speakers. This is not a new concept. It's been already going on since the early days of NanoReg in 2013. Where we developed already an implementation approach to safe by design. And now we include this word, sustainable, and we are busy working on the development of new indicators for sustainability in this framework, which has been also developing in different projects. We are part of those projects. You can see them down on the screen. We have the Harmless Project, Sunshine, and the Go4Nano. We do have our strategies to implement safe and sustainable by design, mainly safe by design with companies. And we have developed a platform, and we work very close with industry. We want to develop solutions that can be implementable. And that's why we are leading a standard and SEN 352 nanotechnologies on the development of a standard for safe by design, which can be actually applicable to industry, because we find that sometimes the world that the European projects follow is not necessarily being followed by down-to-earth SMEs. So we actually try to develop these standards so companies can also be aware of safe by design concepts and implementation strategies. Next one. Here I mentioned nanotechnology. Nanotechnology is at the heart of our work, our everyday work. However, I wanted to have a dedicated slide on nanotechnologies because we are also busy implementing regulatory guidelines to nanomaterials, and we have been there now. We are quite happy to have finalized the skin sensitization guideline 442D, for the application of nanomaterials. This has been performed in collaboration with the Federal Office of Public Health in Switzerland and the GmbH Labs under the Go4Nano project. So this is now out, and it was presented at the OECD meeting in Paris last week. Likewise, we also have a large network of laboratories with expertise in nanomaterial testing, because we found that this is actually a gap, and many companies don't have access to those dedicated labs. And finally, training. We've been doing a lot of activities on training, mainly around the implementation of Safe by Design. We've done it at the industrial settings with industry, also online, and also at dedicated events. And here I highlighted these events also co-organized by BioNanoNet in Venice this year, and we were also present there. And finally, we were asked also to explain why we like to join BioNanoNet. Yeah, like Andreas said, we were members before, and unfortunately we had to leave, and we are back again. We are very happy about this. And our main idea is to expand the sectors that we are covering. I mentioned cosmetics, medical devices, and chemicals. We would like to explore the possibility to also include the nanomedicine field. And of course, we want to benefit with collaborations with many interesting BioNanoNet partners to try to set up new initiatives or maybe co-creation activities with all of you. We also are interested in the EU initiative. You have seen that we are active in some of them, and we would like also to keep up to date with those. And finally, of course, to see if we can explore some opportunities as service providers to all of you. And with this, I would like to thank you for your attention, and I would like to take any questions if you have them. Thank you very much.
Thank you very much, Blanca. Excellent to know again in a nutshell, what the colleagues can get from TEMAS solution. May I ask you one question? Maybe you have answered it already more or less in your expectation slide, but I just want to rephrase it. Which sectors are you mainly focusing on with your regulatory support? You mentioned nanomedicines now in the last slide, but I don't want to answer it for you.
Yes, we would like to get into nanomedicine. We are working very closely now with cosmetics, chemicals, and mainly medical devices. This is our current activity now. And I am a biologist. We have also a sustainability expert person, and also others in the company. We would like to explore the possibilities for nanomedicine as well.
That's perfect. I think you will have a lot of connecting points as well. Also with the platform, with the technology platform on nanomedicine we are running. Super. At the moment, there were no additional questions put into the chat, but I'm sure they will come further maybe later on. Thank you, Blanca, once again.
Thank you. Thanks.
Okay, with this, we come to our third presentation, which makes us very proud to have also Italy on our European country map. That's great. Sheila SADEGHI, a professor in the group of structural and functional biochemistry. I think Blanca will just say, yoo-hoo, perfect fit. In the Department of Life Sciences and Systems Biology at the University of Torino in Italy. I hope I have put it all in the right order. Thank you very much, Sheila, for joining the network. We are all more than curious about what you can present now as the Office of University of Torino. The floor is yours, please.
Thank you very much. So good afternoon, everyone. I am coming from a totally different set of a background that I'm going to talk about, because as you mentioned, I'm actually in a university, and it's totally different compared to the companies and the main projects that the other speakers have put forward. We are a group in University of Torino. I don't have a slide about the university because I'm going to talk about some of the projects that we are interested in within our group. But since everybody mentioned a few words about their institutions. Without the slides, I try to say a few words about the university. This university was founded in 1404. It's one of the oldest universities and one of the largest in Italy, with more than 80,000 students and 27 departments. The department I'm part of, which is Life Sciences and Systems Biology, we are at the interface literally between molecular type of research all the way to plants and also larger organisms like animals, basically. My group, we are a biochemistry group. My background is biotechnology. I actually did my PhD years in Imperial College in London, followed by some research in UCSF in America. And I have literally been always focused on enzymes. And these enzymes could be from a human background, meaning enzymes, which I'm going to talk briefly about in very shortly, or they could be bacterial enzymes, which have nowadays huge applications in biocatalysis. So as I mentioned to you, we are a biochemistry group of research group. We talk about structure and function. So myself, I'm not involved in structure. I'm mainly a functional biochemist looking at different enzymes. And in terms of what I would like to put through in this short time that I have today are different kinds of, let's say, research that we are carrying out in our laboratory currently. And of course, in order to find some collaborators that are interested in the expertise that we actually can offer and have in our research group. So we are mainly involved in drug discovery and drug metabolism, also related to personalized medicine, which I will explain how we are involved in these. And finally, biocatalysis, looking into using these enzymes for synthesis of chemicals, because these enzymes are very highly specific in the reactions that they carry out. And more recently, in terms of sustainability and green chemistry, we have also been using the same kind of enzymes that we have in our laboratory in order to look at biomass conversion and looking at some reactions that during these biomass conversion, our enzymes can actually contribute to, basically. So in terms of drug discovery, a large group of the enzymes that we have produced in our laboratory are coming from the human liver. And these human enzymes as a group are called human drug metabolizing enzymes. And as the name suggests, I won't say any, but more than 80% of the drugs that we actually take should be going through the metabolism and clearance of these set of enzymes that are residing in the liver, in our liver, in terms of clearing these drugs that we are actually taking. So in terms of drug discovery, the pharmaceutical companies, when they actually produce a new chemical entity that they want to test to see if it is viable to then make it into a drug, they actually have to test these chemical entities, these chemical that they have come up with against our set of enzymes, so these drug metabolizing enzymes. Why is that? It is because they have to find out whether these enzymes actually use these drugs and clear them out of our system after they have actually produced the effect that they are supposed to be doing. Or, unfortunately, in some instances, these chemical entities can become toxic going through the reaction of our enzymes, and therefore they could cause problems to the patient. So in terms of toxicity, safety, and also in terms of metabolism, these new chemical entities have to be tested primarily with this set of enzymes from the human liver. And as I mentioned to you, we have been working with these enzymes for the last 20 years, and we have cloned them. We actually have them in their active purified form. And more recently, what we have been doing is, instead of looking in vitro systems to see if they carry out this kind of reaction, we're looking at making high throughput assays. So not just looking at one enzyme, one chemical entity at a time, but an array of the enzymes and more than one drug of interest, basically. And in order to do that, we've been looking at immobilizing these enzymes on surfaces of electrodes and introducing nanoparticles such as gold nanoparticles or graphene oxide in order to enhance the signal of the enzyme. I go one step back. Why electrochemistry and why on the surface of electrodes? Because these enzymes that are from our human liver, they are actually redox enzymes, and during their activity, they require electrons, which can be substituted by immobilizing on the surface of an electrode and inputting the electrons through this electrode system. So currently, we do have these enzymes active on electrode surfaces in the presence of graphene oxide or gold nanoparticles. What we would like to do is to actually introduce them into a microfluidic system and therefore look at an array of these enzymes with very little volume of the compound that the pharmaceutical companies are interested to test, basically. So in this kind of a scenario, we are looking for a collaboration within the consortium, which can offer some kind of a microfluidics expertise in order to miniaturize the system that we already have put in place in order to make a high throughput microfluidic platform in order to profile different chemical entities, and of course in order to see whether they can be viable as a drug that can be administered to humans, literally. In terms of another experimental setup, another research that we are highly involved with is basically, we are in the era of personalized medicine. I'm sure you have all come across. I don't want to bore you with the details, but basically what personalized medicine from our point of view of the enzymes looks like is the fact that we have all of this kind of green and blue and red guys in the population, as you can see on the left-hand side of your screens. And why these colors are different, why they are not the same, is because these same enzymes that I'm talking to you about, drug metabolizing enzymes, they can be present in multiple types of polymorphic variants, we call them genetic variants, literally. And these variants can then, unfortunately or fortunately, give some advantage or disadvantage to the person carrying them. And therefore, one drug might not have the exact same effect in the whole entire population. And therefore, where our enzymes come in is that we have not only the wild type of these enzymes, so what the majority of people in the population are carrying, let's call them the blue guys in the population, but we also have the variants. And therefore, using an array that I mentioned to you before, it would be actually possible in order to say whether the same drug can be taken by different members of the same population, or whether when you go to the doctor, there has to be an array of the chip with the different enzyme on top, and the doctor at the physician within minutes can tell you whether this drug is going to work for you or not, because you are a carrier of a different genetic variation. And therefore, in terms of personalized medicine, the angle we're coming into it is the fact that we have these variants of these drug metabolizing enzymes, and if we can produce a high throughput platform, we can actually, within minutes, actually tell the patient whether they are suitable for taking this drug or not. Because as you can see, I mean, one could say, why do we need to know all of this? If you look at on the right-hand side of the slide, what you can see is that in some instances, people that take the drug, maybe they have a genetic makeup that the enzyme is literally totally knocked out, and therefore this drug cannot be cleared out of the system, and therefore it could cause adverse drug reactions. So after a few days of taking the same drug, which cannot be cleared out of the system, it becomes toxic to the system, and the patient ends up in hospital, and there are instances in which patients have actually died. And I'm sure you have heard about drugs that have been actually taken out of the market, and one of the reasons are these specific adverse drug reactions. At the other extreme, there are people that take a drug, I'm sure you come across as well, saying they had been prescribed this drug by their physician and they don't see any effect. And of course, that is because of the fact that they clear it out of their system much more rapidly, and therefore they see absolutely no effect in taking the drug. And therefore, these patients actually have another genetic mutation. They are another variant of our drug metabolizing enzymes. So in this case, we can contribute by looking at the kinetics, the pharmacokinetics of the drugs that the pharmaceutical companies are coming up with, and we can say whether this drug can be applicable to the majority of the population, or there are certain members of the same population that should not be taking these drugs because of the adverse drug reactions. And again, in this case, we can offer our expertise because we have these human drug metabolizing enzymes, but we would like to join a consortium on personalized medicine, where before checking the drugs in the patients, we can actually be a step before in the proteomics part, let's say, and the metabolomics part to tell upfront whether this drug and the metabolite that it is producing are going to be safe for the patient to actually, with a genetic makeup of that patient, it is going to be safe for the patient to actually take. And finally, the last set of reactions that I would like to talk about very briefly, because I can see I overran, I always talk too much, is by catalysis. And these enzymes that we have, we also have them in the bacterial version, which are much easier to handle and work with, and they carry out very specific reactions like any set of enzymes that you must have come across. And these enzymes, we have shown that due to their specificity, they are by nature green, they carry out green chemistry, and we can produce synthesized steroids, polymers, drugs, indigoids, indirubins, for example, the new coming up kind of derivatives of drugs, as well as more recently we have been able to show that we are able to use in some of the steps, not all the way, but some of the specific steps in the oxidation of lignin containing material in order to get the platform molecules of interest. Our enzymes can actually carry out some of the oxidation steps within these pathways, basically. So in this case, we are also doing biocatalysis, so sustainable chemistry, green chemistry, carried out by enzymes that are highly specific. So instead of doing many chemical steps and purification at every step, the enzyme carries out this reaction as a catalyst, So you require very little amount of the enzyme and in a very specific way. So you get, in majority of the cases, the product of interest without any purification actually required. I stop here. I hope that during the three examples that I have given you, I have mentioned the expertise that we have, which are based on enzymes as biocatalysts, relevant to medicine as well as sustainable chemistry. And I hope to be able to somehow infiltrate some of the collaborations that are already ongoing in the consortiums that are part of the BioNanoNet Association. And thank you very much. If there are any questions, I will be happy to answer them.
Thank you very much, Sheila. It's great to know that newcomers bring in new, fresh ideas and things. You mentioned more or less for all of the technology orientations we have in the association, like for the microfluidics platform, for the nanomedicine platform, as well as for the sustainable chemistry platform, perfect connecting points. And I'm sure there will be lots of opportunities coming up in the future for collaboration. I would have one question about your microfluidic system. Let's say, how major would you, in case of technology readiness level, let's say, assess your technology you would have, or other way around, how easy you find could it be to implement it on microfluidics?
Well, to be honest with you, we have miniaturized our system, and it is possible to use the one-cell microfluidic system in order to look at the turnover of our enzyme immobilized on the surface, but we want to multiplex it. So in terms of a single system, it is doable. We have done it, but it would be very nice if we could have some collaborators which already have the capabilities of multiplexing. So our enzymes are stable on gold or carbon surfaces, and we are talking about surfaces that are require microliters, 1 microliter flow through. And our systems, if anybody's interested, we can actually share what we have already published and how we have set up these electrochemical cells, but we are interested in multiplexing, so we could have the enzymes and their variants in one array, and then the microfluidic system gives the output that in, let's say, in few minutes, we can actually see the reaction of the different enzymes within the array. So for one enzymes, we can do it, and we have miniaturized it. But for multiplexing, we need help, basically.
That sounds very promising for collaboration. And I would say it's also a very good point to look then into the upcoming core topics, where to address maybe in a European project to step forward on that.
That would be fantastic. Super. Thank you very much. Yes.
Okay, I just have one question here now coming in from Blanca. She means, do you take into account enzyme polymorphs?
Yes, exactly. So we are working with the wild type enzyme, so what usually is prevalent in the population, as well as the mutation, so the variant polymorphic enzymes, which are less prevalent in the population. So 1% of the people could actually be carriers. But in some instances, when we say variant, they could be up to 25%. So 1 in every, I don't know, 5 people that are listening could actually be a variant. And therefore, some of the drugs are not good for them to take. And we're sitting here not knowing what kind of a genetic makeup we have, and what kind of a consequence that is going to have on the makeup of these enzymes that are there to metabolize and clear the drugs. So yes, we are talking about also the variants that of these enzymes.
Very good. I think that very nicely answered the question from Blanca, and I'm quite sure that Blanca will have, with her biology background, a lot of further questions. Yes, there's coming the next one. What type of worldwide population do you take into account?
Basically, what we have are, we look at mutations that are already published online from the human genome to say that this is the kind of mutation that they have been seen. And then we clone these enzymes and we look one by one at the reaction with the different drugs that we have available. So one of the, to just give you an example, one of these enzymes actually converts codeine into morphine, and if you and it is prevalent in 25%, and therefore if you are a carrier of this mutation, for example, what will happen is that you cannot convert, you very quickly convert the codeine into morphine, and as you very well know, morphine lowers the breathing and all the functionality of our system, and therefore people have died actually, and therefore FDA as well as the European Safety Agency, they have put out some warnings that, for example, drugs that have codeine in it at the moment, you cannot give it to kids that are, or teenagers under the age of 18, and also for people who are having operations, they have to be tested before they are injected with this. If it is morphine directly, it's okay, but if they are given the codeine, they have to be very, very careful. So this is not a minor thing that can happen. It has huge consequences, and that is why there is so much interest in it. Another example, I don't want to drag on, but is food-drug interactions, and I'm sure if your grandparents or unfortunately yourselves are taking drugs, if you open the leaflet, it says not to be taken with grapefruit juice. Grapefruit juice actually knocks out totally the activity of one of the most important drug metabolizing enzymes, and therefore there is a huge amount of studies currently ongoing to check what kind of food products that we are consuming can affect these enzymes, and the most famous one is actually grapefruit juice. So never ever take a drug with a glass of grapefruit juice.
Wow, really very interesting topic, and Blanca also mentioned that now in the chat. And amazing to get such easy information out of such a, let's say, scientific-based meeting. So I will avoid grapefruit juice anyhow in the future, but I had just a glass of it, so maybe I should stop it here. Super. Thanks a lot, Sheila, once again for a nice presentation, and welcome again also in the association. With this, we are through our official program of that meeting. We hope that all of you really had a very good impression now about what the new members of the association can bring to you. They are very open for any further collaboration, as you have heard. You know, there will come up soon the next versions, draft versions of the working program of the Horizon Europe 2023-2024 program. We will keep you informed, of course. That's one of our services for our members, of course, to keep you informed about what comes up there, and to already start very early to bring together our members for further collaboration. And as far as I hear now from the discussions, maybe Blanca and Sheila will immediately start to collaborate. And I do have in mind, of course, other members in the association, which would perfectly fit for going for personalized medicine, for addressing also maybe lignocellulosic material. That could be also a topic for IVL with John and the colleagues. And I'm sure this intervention today has brought to all of us some ideas for collaboration. Of course, the BNN team will support you with further activities. and we will try to further broaden the network and also to spread the information. You find now in the chat once again the information that our BNN quarterly, which is the public information which goes around the world to approximately 11,500 or 12,000 contacts globally, is an opportunity to spread the word. And of course, please mark your calendar and register already for the next annual event. It's a physical one, which will run on the 27th of September in Tirol, back to back with the Biomedical Engineering Conference, 28th to 30th of September, as well in Tirol in Innsbruck. With this, I want to say thank you to the speakers once again. I hope you all enjoyed the information you just received, and that you will come back if you are interested in any kind of collaboration or interaction, either to the BNN team or directly to the new members. Thank you again for the participation. Thanks also to our internal and external communication team around Simone, Babsi, Beatrice and Caitlin, and of course Melanie from the Alliances and Clustering team for organizing this nice webinar. And by this, I'm happy to now close the meeting and wish you all a very, very nice and hopefully relaxing summer. See you then back at our meetings in September. Thank you.